So hi everyone, thanks for taking the time to attend this talk. Uh, so the title of this talk is a bit long. It's Disentangling Neurodegeneration with Brain Age Gap Prediction Models, Graph Signal Processing Perspective. And this is the same title as our recently accepted paper, which is a tutorial paper to IEEE Signal Processing Magazine in collaboration with Gonzalo Matios and Alejandro Ribeiro. So the content that I'll present today is based on that paper. All right, so brain age gap is the estimate of the biological age of a person, which is estimated from neuroimaging using machine learning algorithms. The biological age of a person can differ significantly from their chronological age because of a variety of factors that are mentioned here, such as environment, genetics, and neurodegeneration. So in this presentation, we'll focus specifically on neurodegeneration because it's easier to see the impacts of this neurodegeneration manifest in the neuroimaging data. Uh, so in neurodegeneration, we expect that the brain age will be significantly higher than the true age of a person. And as a consequence, we can define a metric called brain age gap, which is the difference between the brain age and the chronological age or the true age of an individual. And we expect that this brain age gap will be higher relative to people with neurodegeneration relative to people that are healthy. And we expect that the absolute value of this brain age gap will be, it will somehow reflect the vulnerability or the disease severity in neurodegeneration. So I'll spend next couple of slides just talking about neurodegeneration and how it appears in brain imaging data. So in general, neurodegeneration is the accelerated decline of structure or function of the brain. And we can capture these aspects through regular MRI scans. So in an MRI scan, we can get the images of the brain and we can pre-process them to get some data about the brain. So here I'm going to focus on cortical features, which is where the cortical surface is like the outer shell of the brain, and we can measure its thickness, volume, or area, referred to as cortical thickness, volume, and area here. So all of these metrics show the impacts of neurodegeneration because they significantly decline when there is a disease. So, and this phenomena of decline of cortical thickness, volume, or area is commonly referred to as cortical atrophy. All right, so in this case study, I'll show you how this neurodegeneration is reflected in terms of cortical atrophy. So here I have a data set of 3 cohorts. The HC cohort reflects the healthy cohort, and then we have people with Alzheimer's disease, which is AD, and in between healthy and Alzheimer's disease, there is a cohort labeled as MCI, or mild cognitive impairment. In general, MCI is a precursor to Alzheimer's disease in the sense that individuals are showing some symptoms of cognitive decline, but they do not yet meet the criteria for official diagnosis of Alzheimer's. So we expect that the cortical atrophy would be highest in Alzheimer's disease, and then MCI would show a little less cortical atrophy, and then healthy individuals are your controls against which you compare this. So in these figures, in the first figure, which is labeled A here, we plot the distributions of mean cortical thickness, which is the mean cortical thickness across the whole brain. And you see that the cortical thickness is the lowest, has the lowest mean or median here in Alzheimer's disease. And MCI is intermediate between the healthy controls and the Alzheimer's disease, and then your healthy controls, the controls against which you compare. So this is expected because in like I said, in neurodegeneration, you expect the brain to thin, the brain thickness to accelerate relative to the healthy population. In the next figure, we plot the cortical thickness versus age. So what you see here are three set of points all plotted together, where you see there's a slightly negative slope in how mean cortical thickness evolves with age. So what you expect here is that as a person ages, their cortical thickness becomes thinner or it reduces. Um, and it's a phenomenon of healthy aging as well. The figures C and D, they reflect essentially the brain regions which show the most decline in cortical thickness in the MCI and the AD groups. Now, the key takeaway message here is that just looking at the raw data, that is, say, the cortical thickness data, is not enough to detect neurodegeneration, just because these distributions are overlapping a lot. And furthermore, all of these cortical thickness values are also reducing with age. So it's just that in neurodegenerative condition, the decline is accelerated, but decline is there nevertheless, even in healthy population. So the consequence of this is that we need like machine learning or deep learning algorithms that can detect the patterns within the neuroimaging data that we can then leverage to get a sense of the health status of an individual. Because just looking at the raw data can be very subjective. That is, it may not reflect the true health status of an individual. So brain age gap is a commonly used biomarker in this context, because what it provides us is with a summary statistic of the brain health of an individual. That is, if brain age gap is higher, we anticipate that perhaps that person has some underlying condition that's causing that. All right. So next I'll go over the general machine learning pipeline to evaluate brain age gap. So of course, we do not have a ground truth for the brain age in individuals with neurodegeneration, right? So what we do is we first train the machine learning model to just predict the age of a healthy population. So it's a regular regression model. You feed in the say neuroimaging data, which can be the cortical thickness data, and you want to predict the age of healthy population. And the idea would be that you want this machine learning model to know as much as possible about healthy aging, so that it's able to detect accelerated aging when you feed it to the data that comes from neurodegenerative cohorts. And then the second step is to do some age bias correction. So age bias correction is necessitated because the data that we have might not be the perfect predictor of the age in a healthy population, which might lead to the scenario where if this solid line at the top here represents the perfect fit, but if the correlation between the predicted age and the true age is less than 1, you anticipate a distribution along this dotted line, dotted red line, which means that the people who are younger, this model will tend to overestimate their age, and the people who are older, this model will tend to underestimate the age. So you do some age bias correction to correct for that. And then you have this brain age gap prediction model where you just feed it the data from all cohorts. And then when you look at the distribution of the brain age gap, you anticipate that the brain age gap will be higher in people with neurodegeneration relative to people who are healthy. Right, so I can take any one or two questions here if anyone has any. All right, so I've set up, so there are like thousands of papers that do this. So it's not something that I came up with, but we'll see the problems with this shortly. So before that, let's see like what it looks like in machine learning terms. So what we are talking about is we are talking about training a machine learning model on a large data set, which is the pre-training procedure. So this large data set is that of a healthy population. So we learn something about aging during this pre-training process. And then we apply it on a target data set, which is the neurodegeneration in this case. And we anticipate that this model will transfer its knowledge about aging to detect accelerated aging and neurodegeneration. So makes sense, I hope. So now one key thing to notice here is that usually in transfer learning, we want to preserve the performance of the model. But here we are expecting this model to not perform well in neurodegeneration. Because this brain age gap is essentially the residual of the model, and we are anticipating those residuals to be larger in neurodegeneration to just reflect the accelerated aging. So the distribution here, you can think of them as of the residuals of the model, and you anticipate that the model will not perform well on the target data set, and it will not perform well in a specific way. So it will not perform well in the sense that the residuals will have a positive magnitude to reflect accelerated aging. And hopefully the magnitude will also correlate with the disease severity or the status of an individual. All right. So in order to realize this, we want to have a choice of a machine learning model in this pipeline that actually leverages the data set in some principled manner so that it can detect those patterns of accelerated aging, right? So most of the approaches in this application, they focus on achieving the perfect performance in the pre-training step, because that is something that you can control. You have a solid ground truth, which is the age of a person, and you can try to achieve as much as perfect prediction as possible of age on healthy population. And then we call those as performance-driven approaches because they focus just on the performance of the pre-training step. And the drawback or the restriction or the constraint there is that it does not guarantee a meaningful brain age gap in the target population, which is the neurodegeneration. So in the pre-training, we have not seen this data at all. And since these approaches just focus on age prediction, we cannot guarantee that the model performance will fall apart in the target population. In fact, if it does not fall apart, I mean, the model is doing its job. It's just that it's not useful for a clinician or a patient. So this is one of the methodological obscurity that's there in this standard brain age gap prediction pipeline. That is, we can use these powerful neural networks to predict age in healthy population. It's just that the performance here has little to no correlation with the brain age gap prediction in the target data set, which is the neurodegeneration. So since we do not have this correlation, essentially we have a large number of studies that cannot address this particular gap that's there in this pipeline. So I'll put this gap in this abstract way. So what I mean to say is that the performance in pre-training does not dictate meaningful brain age gap or residuals in the target population. All right, so after identifying this gap in the literature, we came up with like some principles that we need to incorporate within this brain age gap prediction pipeline. So we argued that we should focus on the residuals of the machine learning model and not the prediction performance, because that's what matters in this application. Residual is the brain age gap, and we want it to behave in a certain way in the target population. The second thing is that we should focus more on the qualitative evaluation during pre-training. That is, we should not, rather than focusing on the performance of the model and the age prediction on healthy population, we should rather try to surmise what does the model learn during pre-training on this population. The third aspect is the interpretability or explainability of brain age gap. So here we argue that we should try to find factors that are contributing to this elevated brain age gap in the neurodegenerative conditions. And this can also help validate uh, uh, the biological meaning of the observed brain age gap. So, uh, in the sense that each of these neurodegenerative conditions have some anatomical signatures where certain brain areas are impacted. So, if you show a clinician or a patient that this inferred brain age gap is driven by these particular set of regions, then perhaps it's easier for them to trust it more in practice. And lastly, we want this prediction pipeline to be generalizable to diverse target populations, by which I mean that there are various types of neurodegenerative conditions that show accelerated aging. So we want this pipeline to be generalizable wherever applicable. All right, so now we in our paper we proposed that we can use a VNN model, which is short form for a covariance neural network, to predict brain age gap, and I'll describe VNN in a little bit. But the key things to note here is that the VNN model leverages the covariance matrix derived from the neuroimaging data, and it processes the input cortical thickness information according to this covariance matrix, and then the rest of the pipeline remains the same. And one key advantage here is that we can also talk about the anatomical interpretability. That is, we can identify what is contributing to this brain age gap in the downstream. So here the brain regions labeled in red are the ones that we can conclude to be contributing to this elevated brain age gap. So essentially this is your brain age gap. This becomes its anatomical signature, which can then be verified by a person with, say, biological expertise. So in the sense that they can confirm that these regions make sense for a particular diagnosis. And lastly, we can also talk about how this covariance matrix is leveraged in terms of its eigenvectors to generate these patterns, and more specifically, how it's leveraged differently in healthy population and in people with neurodegeneration, so that these sort of patterns appear that lead to this elevated brain age gap. All right, so here, like I said, we will work with the covariance matrix, which is like a network structure of the brain. And in general, when we look at the network structures in neuroscience, we club those approaches together under the domain of network neuroscience. So here are like two examples of networks that are commonly used. One is anatomical covariance matrix, which you can evaluate as a general sample covariance matrix on neuroimaging data. And you can also have a functional connectivity matrix, which just represents the correlation between the time series at different brain regions. And the motivation here is that the data at different regions of the brain, it has significant redundancies. So these networks essentially can capture those structure within the data. And secondly, when we talk about accelerated decline in these, say, cortical thickness or any other features, the result of that is that these networks are also impacted. So the impact on those networks is also itself a biomarker or a indicator of the neurodegeneration. Now, one thing you might notice here is that both of the networks that I talked about are essentially covariance matrices or correlation matrices. Uh, uh, and if you want to apply machine learning over covariance matrices, you can do that both in a traditional way or a modern deep learning way. In a traditional way, you would do it through, say, for instance, principal component analysis, where the advantage would be that you would get a model which is interpretable and suitable for low data regimes. The disadvantage would be that this model might not be stable, or it might not lead to reproducible results. And then you have deep learning approaches like graph neural networks, where you consider this covariance matrix as a graph, and then you will probably get an improved performance. All right. So in our method, we of course use the covariance matrix as this data-driven graph. So it's a fully connected graph. Here's just an example of covariance matrix consisting of 6 features. So each of those features form the nodes of this graph, and it's evaluated in a regular fashion as a sample covariance matrix. And when you look at it in terms of the brain, what it represents is that the nodes of this graph are essentially the brain regions, and the connections between these different regions in this graph represent the covariance values or the pairwise covariance values. All right, so we want to exploit this structure in a principled manner. And the way to do that is graph signal processing. So we know that signal and information processing is about exploiting the signal structure, and graph signal processing generalizes that aspect to processing the signals over graphs. So there are lots of tutorial papers or review papers that study this aspect. Some of them are shown in the slide. So essentially through graph signal processing, we want to process a graph signal. So the basic definition of a graph signal is that it's defined on the vertices of the graph, and it maps each of those vertices to some data point, which could be a scalar or a vector quantity. So we can represent this graph signal as a vector in this way, and each of the elements of this graph signal are associated with distinct nodes over which you project the data in the graph signal. So I would say so it's easy to see that the cortical thickness features that we talked about earlier, they are graph signals, because we are talking about the cortical thickness of different brain regions. Each of the brain region forms the node of the graph, and the cortical thickness data forms these elements of the graph signal. And then graph signal processing, the fundamental information processing module is a graph filter. So in this slide, we have the representation of a graph filter for a general graph representation, matrix representation S. So this filter generates the output Z by this polynomial, through this polynomial function defined over the graph representation S, and which is aggregated together through these filter tabs or scalar values HK, which are referred to as filter tabs. The impact of this mathematical expression is shown in just figure at the bottom, where depending on the power of S here, we essentially combine the or aggregate the information in different sized neighborhoods across a node, and then combine them together. The combination or aggregation is dictated through this scalar values HK, which are actually the learnable parameters that you find that you learn from the data during training. So we can of course define this graph filter on a covariance matrix in a straightforward way. You just replace S with C hat, which is your estimate of the covariance matrix, and you get the same implementation. So the interesting thing here is that when you do a theoretical analysis of this graph filter on the covariance matrix, you get some observations that are more meaningful than that you would get through just a regular graph filter. So if you apply this eigen decomposition of covariance matrix C, you reach this expression at the end, where you can see that the term on the right in this expression, which is B hat transpose X, B hat are the set of eigenvectors of the covariance matrix. So this term is actually the principal component analysis. So that's how it's defined. And in the middle, you have this polynomial over the eigenvalues of the covariance matrix. So essentially these weights hk are manipulating these eigenvalues in a certain way that aligns with the learning objective. And then you have a v hat towards the left, which just projects everything back into the space of the data that we have. So because of this conceptual equality of a graph filter over covariance matrix and principal component analysis, in other works, we pursued this idea further to develop some theoretical results. And because of that, we call this we study it separately as a covariance filter, that is a graph filter applied on a covariance matrix. All right, so this is our fundamental information processing module, that is a covariance filter. So we know that this filter can learn only linear representations. So to accommodate nonlinear representations, we just form perceptrons out of it by concatenating it with the pointwise nonlinearity, like a ReLU or a sigmoid or whatever. And then with this perceptron, you can generate deep learning models of any arbitrary architecture. So, so then we have this covariance neural network formalized now, where the input data X is processed through some combination of these perceptrons. Each perceptron consists of a covariance filter, and we get some output. So here we can also conclude that just because of the theoretical analysis of this covariance filter, we can further conclude that this covariance neural network must be exploiting the eigenvectors of the covariance matrix in some way in order to generate the output representation. All right. Now there are so various properties of these VNN models make them appealing for neuroimaging data analysis. One of them is connections with PCA. What it leads to is transparent outcomes by leveraging the spectrum of the covariance matrix. I won't say it's fully transparent because we have nonlinearities in between, but there is still some sense of transparency in the sense that we can tie the outcomes of a VNN to how the eigenvectors were exploited. And then we have two theoretical properties. One is that of stability, where we can talk about the reproducibility of the outcomes in limited data settings. This is a theoretical paper that appeared in 2022. And then we can also claim transferability, where we can show that these models are robust to the choice of the structure of the cortical thickness features. So I did not talk about this in much detail, but I'll try to cover it in a slide towards the end if we have time. All right, so let's first see like how this model does on the pre-training task. So recall that for brain age gap prediction, we want to train this model to predict the age of healthy population. So we have the input as the cortical thickness data. We want to form the estimate of the age, and our baseline is standard PCA regression. So in this plot at the bottom left, what we see is that the point that's marked with the blue arrow, that's the point of comparison of the test performance of PCA-based models and a VNN model. So VNN, of course, outperforms these PCA models. And what you see on the left and the right is that then you re-estimate the covariance matrix with a smaller or larger number of samples. So your covariance matrix is slightly perturbed relative to what you have during the training process. And what you get is that if you keep the weights of the PCA model and the VNN models the same, then just switching this covariance matrix leads to significant degradation and variation in the performance of PCA-based models, which is not the case for a VNN model. So essentially the performance that you get, it's robust to the number of samples that you use to estimate the covariance matrix. So this is important because we do not want the performance of the model to be overfit on the particular training data set that we have. So if it's robust, then we can also claim reproducibility of the performance. So all right, so we have this pre-trained model which has its own advantages, and then we see how it leads to a meaningful brain age gap. So what we did here was that we set up this regression model with an unweighted readout function. So you process the input cortical thickness data, you get the representation that's of the same dimensionality. And instead of putting like a linear or a non-linear learnable readout function, we just have this unweighted, that is, we just aggregate the values here and we get the prediction of the age. And it helps us in the way that we have to apply this VNN model on a target population, right? So what we anticipate is that when the performance will fall apart, we can keep track of where the performance falls apart in the output representation of this VNN model. And if it falls apart consistently in a consistent way across the whole neurodegeneration data set, then we have an anatomic signature about that health condition that's leading to an elevated age prediction. So we look at the anomalies. So essentially what we do is we look at the distribution of the representation for the healthy individuals. We look at the same for the people with neurodegeneration, and we do some simple statistical analysis like ANOVA or ANCOVA or whatever, and get these plot of brain regions that are showing which regions might be leading to an anomalous age gap prediction in the downstream. And it's easy to show mathematically that whatever anomalies that you observe here, they basically get propagated to this brain age gap output. So so we have these anomalies in the VNN output. They get transformed through this age bias correction model, which is a standard linear model. And those anomalies basically just contribute to this elevated brain age gap in the downstream. And in the intermediate step, you can look at the anomalies at the representation of the VNN, and those we refer to as the regional residuals, which can be termed as the contributing factors to the brain age gap, the downstream. We can also tie this to the eigenvectors. I don't think I have the slide here, but we can also tie the VNN representations with the eigenvectors of the covariance matrix in a post hoc way, where you try to see which eigenvectors these representations align to. That tells you about how those eigenvectors were exploited differently. All right, so next I'll describe some results. So we had the data set. So we had this OASIS 3 data set, which is one of the publicly available data sets of for Alzheimer's disease. So the table summarizes the demographic information for that data set. So we had this HC group, which is the healthy group, ED group, the Alzheimer's disease group. And for each of the person diagnosed with Alzheimer's, we also had their clinical dementia rating value, which is the which is the value provided by the clinician in the when they gauge the severity of the disease clinically through various cognitive tests. So so we see that the brain age gap is elevated in Alzheimer's disease. We see some anatomical patterns assigned to it. And these patterns are very much consistent with the most significant regions in Alzheimer's disease. So I want to emphasize here that in general, you would observe cortical atrophy across the whole brain. That is cortical thinning across the whole brain in Alzheimer's disease. But the regions that are highlighted here are among the ones that where the disease tends to evolve or its evolution starts in the brain. And then these brain age gap values, they also correlate with the values of the CDR, which is the rating given by the clinician just suggesting that these brain age gap values do have some meaning. Secondly, we can look at how different eigenvectors were exploited during this brain age gap prediction, and we do see some significant differences there. So here, inner product metric on the Y column is the essentially the inner product between the representation of the VNN and the eigenvectors of the covariance matrix, which gives us some idea into how those representations generated by the VNN correlate with the eigenvectors. And we find that those correlations can be significantly different for healthy individuals and people with Alzheimer's disease. Just gives us some insights into how the anatomical interpretability for this brain age gap was generated. And we can, of course, extend this to other diseases. So one of the extensions here that I pursued here is that of frontotemporal dementia. As the name suggests, this kind of disease, it impacts more the frontal regions and the temporal regions of the brain. So again, we see the brain age gap distributions that make sense. And we also see these implicated brain regions that also make sense for this disease. And again, we see some anomalies in the how the eigenvectors were exploited, which explains how the results on the left here were generated. All right. So before I end, I also want to briefly mention the transferability aspect of the VNN models. So something that's of interest, that's something that's interesting about VNN models is that they can process the data set of any arbitrary dimensionality, which means that you could have your brain data curated according to these different maps, which are dividing the brain surface into different number of brain regions. And you can get the consistent results using these data sets with the same VNN model. So here, the point to note is that the covariance matrix for such data sets would be of different dimensionalities. Still, the same VNN model is able to exploit these different covariance matrices to get a consistent result. And that's something shown in the slide. So we set up the brain age gap prediction pipeline on a data set that consisted of 100 cortical thickness features across the whole brain, and we got the results in the first column here. And then we deployed the same VNN model on data set with 300 features and 500 features and got more or less consistent results. So this just shows that the results that we got were generalizable and robust to how we divided the brain surface. All right, so I'll come towards the end of my presentation. So brain age gap prediction models are promising models for clinical usage because they work as pre-trained models that we can use to infer brain age gap in different health conditions. And through the use of covariance neural networks or VNNs, we are able to propose a principled approach to this particular problem, where the key advantages are we can talk about how this brain age gap is anatomically interpretable and explainable. And lastly, the transferability of VNNs also provides a way to cross-validate the results that we get across different brain atlases. Again, adds a metric of confidence to the reported results. All right, so this is where I'll end, and thank you for your attention.